क्योंकि ये फेसबुक पे जाना है। चलिए, so it is asked define genetic code, right? What are its characteristics or what are its salient features? So till 2016 ये सीधे सीधे question, like direct question was asked. Afterwards, after 2017, when application based questions were like the percentage increased for application based questions, the questions were being asked generally. Genetic code is unambiguous in nature, genetic code is universal in nature, justify it. So it answer always is the same, but only the way the question is being asked, it's a little bit twisted, and answer is given. Answer you have to give on the basis of question. So the number of participants which are attending the class, so I will start with the discussion process, so that because when all the peer are present in the class, and with that flipped learning, it is very easier to catch the concepts. So if we have to give the definition, what is genetic code? अपने आप को please unmute yourself and then you can answer one by one. What do you mean by genetic code? If the simple definition of 1 mark is being asked, generally in 1 mark definition as such will not be asked because you are having MCQs, but for 5 marks questions, there is a possibility the question is broken down as 2 marks, 2 marks and 1 mark. There the definition can be asked, so we need to prepare answer from every aspect. Anji, genetic code. Imnandan, unmute yourself, bache.
Information that is transferred during translation.
Information that is transferred during translation is a simple thing. Otherwise, we will say a code of 3 nitrogenous bases encoding for genetic information is termed as genetic code. A number of scientists were involved in it, like Mr. Hargobind Khurana, Mr. Neil, Mr. Gamow. So it was not like it is not the necessary only the biology candidates were involved in its discovery. Mathematicians or the physics person or biochemists were also involved. So George Gamow, like he gave the rule 4 raised to power 3, means there are the 4 nitrogen bases, and those nitrogen bases are adenine is there, guanine is there, cytosine is there, and uracil is there. Because we are encoding the information, and when we are talking about encoding the information, means we are looking forward from transcription to translation, where the genetic information will be expressed in the form of enzymes, in the form of proteins. So these are the 3 nitrogen bases. So these 4 nitrogen bases, they are combining with each other in 3 ways, so that their total number of codons are 4 into 4 into 4, and this is equal to 64. So it means in the genetic code there are total 64 codons, and each codon is in the form of 3 nitrogen bases. So a checkerboard was given, and in this checkerboard, what was there? These 4 nitrogen bases were here. These 4 nitrogen bases were written here, like adenine, guanine, cytosine, or uracil. And with the third position, the checkerboard which was there, adenine, guanine, cytosine, uracil. Again, there were adenine, guanine, cytosine, and uracil. So when the codon of 3 nitrogen bases were involved, so how was it? A means the nitrogen base present at the first position, nitrogen base present at the second position, and remains constant, but there is a change only for the nitrogen base present at the third position. मतलब पहले में क्या बना? A, सेकंड में क्या बना? A, G, third में बना C, and the fourth one is A and U. So in this way, the 4 nitrogen bases, they are combining in the 3 ways, and ultimately there are 64 codons are there. Out of these 64 codons, the functional one, they are only 61 codons. Means they are coding for some kind of proteins. But the 3 codons were figured out which were not coding for any kind of protein. They were having no role in translation. And if there was any anticodon present for them, during translation process, if they are present on the messenger RNA, there is no anticodon given by transfer RNA. So these codons were called nonsense codon, means stop codons. They are not carrying on this translation process. Can any one of you unmute yourself and let me know about those 3 codons which are not coding for any protein? Anyone?
UAA,
Very good.
UGA.
Yes. UAA, UAG, and UGA. Right, bache? So these are the 3 stop codons which do not code for any codon. So till here you are clear about the basic information of genetic code. Now we need to know what are the various characteristic features of these codons. So now very rare chances, आपके वाले टाइम में चांस हो सकता है कि ज्यादा सीधा क्वेश्चन आ जाए, बिकॉज इफ यू सी योर दिस वन, क्या कहते हैं इसको, अपना सिलेबस का देखो तो उसमें इन्होंने थोड़ा सा बोला हुआ है कि ये एप्लीकेशन बेस्ड क्वेश्चन थोड़े कम हैं, अंडरस्टैंडिंग बेस्ड क्वेश्चन जो हैं वो थोड़े से ज्यादा करते हैं. तो अगर आपसे ये पूछा जाता है कि नेक्स्ट व्हाट आर द कैरेक्टरिस्टिक फीचर्स, सो व्हाट विल यू से, व्हाट आर द कैरेक्टरिस्टिक फीचर्स? कौन कौन से कैरेक्टरिस्टिक फीचर्स आपको पता है कि ये होते हैं? हाँ जी, एनीवन?
मैम जेनेटिक कोड इज डीजेनरेट.
इट्स अ डीजेनरेट. चलिए हम पहला इसका लिख लेते हैं. आगे हाँ जी बच्चों,
यूनिवर्सल.
इट इज यूनिवर्सल. वेरी गुड. सेकंड वन
Universal.
यस. नेक्स्ट जपराज, आगे बच्चे.
मैम इट इज अ ट्रिपलेट.
Initiated.
पार्डन बच्चे.
Initiated on.
There is a initiator codon.
Initiator codon.
Initiated.
Fine. Initiator codon.
मैम इट इज अ ट्रिपलेट.
They are always present as the triplet, means 3 nitrogen bases, or they are always
Function as stop codon.
punctuationless. ये हमेशा punctuationless है. इसमें किसी तरह का full stop, किसी तरह का comma नहीं होगा. और कोई अगर हम मिस कर रहे हो characteristic feature, is there any other feature? They are unambiguous. राइट. चलिए अब मेरे को ये बताइए कि इसमें हमें सबसे इजी है यूनिवर्सल कोडोन. यूनिवर्सल मींस यूनिवर्सल ट्रुथ हम कहते हैं. ऑनेस्टी इज द बेस्ट पॉलिसी. ऑल वेल, ऑल एंड्स वेल, ऑल क्या कहते हैं उसको, ऑल इज वेल दैट एंड्स वेल, समथिंग लाइक दीज आर द यूनिवर्सल ट्रुथ which never changes. So universal means these codons are throughout in plant kingdom and animal kingdom. They are same in any eukaryote, prokaryote, except a few bacteria, those prokaryotes, or for some mitochondrial genes. So give one example. If I am saying this is universal, let's say U, it codes for
Phenylalanine.
U if present in your body, in my body, in a fungus, in a plant, it will always code for phenylalanine. So that's why it's termed as universal. अच्छा, एक इजी वाले के पास चलते हैं. जपराज ने बताया था ट्रिपलेट means they are always present in the 3 nitrogen bases. You will never see कि कोडोन ऐसे लिखा जाए. 4 कभी नहीं होंगे. You will never see कि ये 2 लिखे जाएं. It will never happen. लेकिन इसमें हमेशा क्या होगा, या ऐसे लिख दिया UUU, या आ गया आपका UA, U आ गया, या GC, A आ गया, somewhere whatever the thing. ये इस तरह से हमेशा क्या होंगे, ट्रिपलेट होंगे. एक और easier one की तरफ हम चलते हैं. The easier one, it is called initiator codon. Which is called initiator codon? जिसके बिना translation process cannot be started. What's that?
AUG.
Very good. AUG. And do you know this works as a dual function? And dual means 2 functions. One function already is there, initiator codon. So what is the second function?
Codes for methionine.
Very good. It codes for methionine. Right? It has the 2 roles. One person is playing 2 roles. Okay. So universal is also done. It's applicable everywhere. Initiator ho gaya, triplet ho gaya. Next we are coming to the another easier one, which is called punctuationless. Means the codons on these DNA, they are present. Let's say UUU, UGA, AAA, AG, C, like that. So understood that they are read as a 3-1. 3 U को अलग से पढ़ेंगे, UGA को अलग से पढ़ेंगे, AA को अलग से पढ़ेंगे, AGC को अलग से पढ़ेंगे, but there will be no comma, no hyphen, no gap, nothing at all. Right? This is like our email ID or passwords, इसमें हम gaps नहीं डालते. So coming to the first and the bottom one, it is called degenerate. So degenerated means, let's ask Harman. Harman की English सबसे बढ़िया है. So Harman, what is the meaning of degenerate?
It can simply degenerate itself, is what I can say.
Right. So yes, you are absolutely right. And in this, what is there? Sometimes some of the codons, जैसे अपना checkerboard देखो, ये आपका जो table 6.1 दिया हुआ है, so here you will see some of the codons you will see, they are coded by more than one type of codon. Let's take the first 2 examples given, phenylalanine. अगर यहाँ पे इन्होंने दिया है UUU और एक सेकंड इन्होंने साथ में दिया हुआ है UUC. So you will see in your textbook, both are called as phenylalanine. Means amino acid is one, but the codes are of 2 different categories. UUU as well as UUC, both are coding for phenylalanine. Then it is termed as degenerate. So one last I have given you here, which is called unambiguous. What is its meaning? Unambiguous.
Clear without any
Without
confusion stuff.
any confusion, they are clear. Means if phenylalanine, it is coded by UUU, I mean this UUU will code only for phenylalanine, it will not code for any serine. So if somebody will ask you what does it code for, ये नीचे लिख देती हूँ unambiguous के पास कि UUU किसके लिए code करता है, तो our answer is it is for phenylalanine. Somebody is asking you, is it coding for aspartic acid? Is it coding for histamine? Is it coding for methionine? No. Our answer is very clear. It is unambiguous in nature. And total there are 64 codons and there are 3 stop codons and 61 they are termed as functional codons. Clear Japraj? तो Japraj का लगता है कैमरा हैंग हो गया. So
Clear, ma'am.
okay बच्चे, fine, thank you. अगला बोलो टॉपिक.
Can I say?
Yeah, please.
Translation?
Please.
Translation.
Translation,
Can
yes.
you please explain translation?
Sure बच्चे. So translation process. अच्छा, अब ट्रांसलेशन में क्या हो रहा है भाई? सबसे पहले हमें पता होना चाहिए व्हाट इज सेंट्रल डोगमा. व्हाट इज दैट सेंट्रल डोगमा?
डीएनए से आरएनए बनता है, आरएनए से प्रोटीन.
Right. First of all, DNA to DNA means replication. Then the copy of DNA formed and it is further forming RNA, and then from RNA to proteins. We know there are three types of RNA. One is called messenger RNA. Another one is termed as ribosomal RNA. And one is termed as transfer RNA. If these RNAs are the part of, they are the RNAs of prokaryotes, there is only one RNA polymerase enzyme. But if these RNAs are formed in a eukaryotic body, the three different types of the RNA are required for these three different types of RNAs. And moreover, in eukaryotes, this transcription process is tricky. I mean, it's a complex. Number one, due to the, you can say, due to this post-processing, because the RNA which is formed, it's not mature to do the function of translation. And second thing, because of the presence of three different types of enzymes. Now next, when we are moving ahead towards the translation. So in the translation process, what are the requirements? What do we need? First, we need these three types of RNAs. Second thing, we need enzymes. Here is the need of enzymes, as well as there is the need of energy, so that these RNAs can be energized. Now everybody knows about the tRNA. Its structure is like clover leaf model. Means clover leaf which has 4 lobes like structure, and it is also having 4 lobes. We are the first lobe which accepts the amino acids, right? And one is which is attached with the enzymes. One lobe where the ribosomes are attached, because you know ribosomes are the protein factories, and the bottom one where the corresponding anticodons are shown. Clear, bache? So whenever any amino acid is attached with the transfer RNA, a complex is formed which is termed as amino acyl transfer RNA. So this is the complex is formed. Means amino acid जो हमें चाहिए वो transfer RNA के साथ जुड़ गया. And this is a highly energy consuming process, and that energy will come from our energy currency of the cell, that is termed as ATP. And ATP is not consumed like 3 phosphates are always consumed. Only one phosphate is consumed at a time. So it means one phosphate, when it is consuming, when it is being used, so we will say AMP is attached with this complex and 2 inorganic phosphates are released. So the complex here is formed, which is called amino acyl transfer RNA complex, and that is energized. And this complex is formed with the help of an enzyme. Please note down the enzyme name, amino acyl transfer RNA synthetase. So this was the first thing, which is the formation with the help of ATP, with the help of amino acid, with the help of transfer RNA. Now 3 things are consumed here. Is it clear till here? Is it clear?
Yes, ma'am.
Now
Yes, ma'am.
मैं जरा इसको ना क्लियर कर रही हूँ इस ड्राइंग को. चलिए नेक्स्ट सेकंड स्टेप. अब हमारी बारी आती है जो हमने कहा राइबोसोम्स की. So ribosomes, which are called protein factories, because this translation process is taking place in the cytoplasm of the cell, right? So ribosomes are present in the cytoplasm of the cell, and we have 2 subunits of ribosomes. One is called 60S, means बड़े वाला ribosome की subunits होती है, and the second one is termed as 40S, right? So these 2 subunits, generally they are separate. but at the time of protein synthesis, with the requirement of magnesium ions, join with each other. ये join ही सिर्फ होती है at the time of protein synthesis, right? Now the third role of messenger RNA. We have studied the role of transfer RNA. We have studied the role of ribosomal RNA. Now we are going to study the role of messenger RNA. So messenger RNA works with the help of enzyme which is called RNA polymerase. RNA polymerase brings this messenger RNA in this groove. ये जो उनके बीच में groove होता है छोटे राइबोसोम के पास. So in this groove, messenger RNA, it is brought with the help of the RNA polymerase. और इसका काम क्या है? इसने रीड करना है कि ट्रांसफर आरएनए ने कौन सा अमाइनो एसिड लिया और कौन सा उसके पास एंटीकोडोन है, so that the corresponding codon it can carry. Please note this point, क्योंकि इस राइबोसोम के अंदर ये ट्रांसफर एनर्जाइज्ड जो ट्रांसफर आरएनए है ना, ये यहाँ पे आके जुड़ जाता है. ये यहाँ पे आके जुड़ गया. अब इसने जो अमाइनो एसिड यहाँ पे रखा हुआ है उसके कॉरेस्पॉन्डिंग इस नोड के पास क्या होते हैं? एंटीकोडोन्स. बिकॉज कोडोन और एंटीकोडोन में कॉम्प्लीमेंट्री बेस पेयरिंग टेक्स प्लेस विद द हेल्प ऑफ हाइड्रोजन बॉन्डिंग. And we just studied genetic code. What is the initiator codon? What is the initiator codon?
AUG.
AUG. So it means messenger RNA, it will bring here which one? AUG. And the amino acid which is attached here with transfer RNA, that will be also methionine. जो इसके साथ जुड़ा होगा उसी को रीड करेगा हमारा आरएनए. अगर आरएनए यहाँ पे एयूजी लेके आ रहा है तो एंटीकोडोन ट्रांसफर आरएनए के पास कौन सा होगा? अगर आरएनए यहाँ पे बच्चे एयूजी लेके आया है तो ट्रांसफर आरएनए के पास यहाँ पे कौन सा होगा? ए के साथ कौन बॉन्ड बनाएगा? यू. We are not talking about DNA. We are talking about RNA here. We are talking about uracil. Thiamines with DNA. वो replication के साथ खत्म हो गया वहां पे जाके. जब हमने ट्रांसक्रिप्शन और ट्रांसलेशन की बात करनी है तो we are talking about uracil here. तो ये जो AUG यहाँ पे आया है मैं यहाँ पे लिख रही हूँ so that it is clearly visible. और एंटीकोडोन जो मैं बात कर रही हूँ ट्रांसफर आरएनए के पास यहाँ पे क्या होगा? U होगा, A होगा और नेक्स्ट क्या हो जाएगा? यहाँ पे C हो जाएगा. एंड देयर विल बी द कॉम्प्लीमेंट्री बेस पेयरिंग बिटवीन दीज एंटीकोडोन्स. जो ट्रांसफर आरएनए की नोड पे प्रेजेंट होंगे उनको हम कहेंगे एंटीकोडोन्स. जो मैसेंजर आरएनए लेके आएगा उनको हम क्या कहेंगे? कोडोन्स. इज इट क्लियर एवरीवन? नाउ अब ये तो बनी हमारी एक बॉन्डिंग बनी. We are studying protein synthesis और प्रोटीन बनते हैं पॉलीपेप्टाइड को कहते हैं. So what is a polypeptide? What is a polypeptide? Anyone? Yes बच्चे, what is a polypeptide? केमिस्ट्री में भी पढ़ा होगा ये आपने. पॉलीपेप्टाइड. पेप्टाइड एक बॉन्ड बनता है. पेप्टाइड बॉन्ड तब बनता है व्हेन टू अमाइनो एसिड कंबाइन विद ईच अदर. यहाँ पे लिख रही हूँ एक अमाइनो एसिड AA, इसके साथ जब दूसरा अमाइनो एसिड बनेगा जुड़ेगा, व्हेन टू अमाइनो एसिड जॉइन विद ईच अदर देयर इज द एलिमिनेशन ऑफ वाटर and both the amino acids after eliminating water they form a bond in between them which is termed as CONH. In your chemistry language we call it as amide bond and in bio language I am calling it as peptide bond. When this is one peptide which is formed by joining two amino acids, if there is a third amino acid also joining here, so then what will happen bachche? How many peptide bonds will be formed? If we are joining 2 amino acid, 1 peptide bond is formed. If we are joining 3 amino acids, how many peptide bonds will be formed?
2.
2.
2,
If we are joining 4 amino acid, how many peptide bonds will be formed?
3,
3.
We are joining 5 amino acids, then how many peptide bonds are formed?
4.
4. It means when one bond is formed, we said monopeptide. When two bonds are formed, we said dipeptide. When there are 3 peptide bonds formed, we said tripeptide. But when there are 4 peptide bonds or more than that formed, what will we say?
polypeptide,
Polypeptide.
Polypeptide. And when so many bonds are present in one chain, so what that chain is called? A chain,
polymerization,
a chain having polypeptide bonds, what that chain is termed as? क्या कहेंगे हम उस चेन को? पॉलीपेप्टाइड चेन. And polypeptide is the another word for proteins. Proteins भी क्या है? They are proteinaceous. Proteins जो हैं वो किससे बने हैं? अमाइनो एसिड से. और ये जो तीन नाइट्रोजन बेस का एक कोडोन आपके सामने है, दिस इज व्हाट? दिस इज अमाइनो एसिड. इसका मतलब है अगर हमने ये एक रिएक्शन करी थी UAC और इसके साथ AUG, अगर इसके साथ कोई और सेम जुड़ता है या डिफरेंट जुड़ जाता है UAC और AUG, तो इसके साथ तो बेटे बन गया एक बॉन्ड यहाँ पे, फिर यहाँ पे जुड़ेगा तो और बॉन्ड, यहाँ पे जुड़ेगा और बॉन्ड, यहाँ पे जुड़ेगा तो अगला बॉन्ड बनेगा. एंड दिस चेन इज टर्म्ड एज पॉलीपेप्टाइड चेन और इट इज कॉल्ड प्रोटीन फॉर्मेशन. क्लियर हुई है बात? इज इट क्लियर एवरीवन? नहीं क्लियर हुआ
Yes,
तो
ma'am.
यस, अगर क्लियर हुआ है तो यस बोलो. बेटे ये जो राइबोसोम्स होते हैं ना,
Yes, ma'am.
there are many ribosomes present in cytoplasm. एक राइबोसोम नहीं होता, बहुत सारे राइबोसोम्स होते हैं. And these ribosomes have their transfer RNAs, and these transfer RNAs are carrying amino acids. So when these amino acids are joining with each other, the peptide bond is formed and it is exiting from this ribosome to the cytoplasm, and more amino acids are entering. So in this way, the polypeptide chain or protein chain is formed. But always there is the end, termination of any process. कभी ना कभी वो protein synthesis बंद तो होगी, नहीं तो unlimited चलती जाएगी. So what is the termination? When this messenger RNA is having any kind of the stop codon, कहीं भी यहाँ पे वो UAA, UGA या UAG कोई भी एक कोडोन आ गया, तो that will not be having any anticodon for that transfer RNA. So here it will be the end of protein synthesis. यहाँ पे खत्म हो जाएगी protein synthesis. And we know that the proteins are building blocks of our body, because whatever we do, whatever the conversions are taking place, metabolic reactions are taking place, they are all because of these enzymes, and enzymes are proteinaceous in nature. Whatever the immune system we are having, the immunity due to the immunoglobulins, they are also due to these proteins. So that's why we always focus, even in our diet, the proteinaceous diet. We always say we should not focus if we on carbohydrate rich diet. Always focus on proteins diet, because they are the building blocks of our body. Though naturally these proteins are formed, but additional source of proteins we also take care in our diet. So this was protein synthesis. हरमन क्लियर हुआ बेटे आपको?
yes ma'am,
इसका हम
thank you so much.
रीकैप करते हैं हम दोबारा से इसका. सो देखो बेटे, सबसे पहले आपको पता होना चाहिए डेफिनेशन ऑफ ट्रांसलेशन. 5 मार्क्स का अगर ये पूरा का पूरा क्वेश्चन आता है, तो ट्रांसलेशन की डेफिनेशन क्या होगी? द फॉर्मेशन ऑफ प्रोटीन्स फ्रॉम द एनकोडेड कोडोन्स ऑफ नाइट्रोजन बेसिस. So that is the translation. Then second step you need to mention, I'm telling you this is for 5 marks question, you need to mention the machinery required, means the tools required. तो tools required में आपके पास क्या होने चाहिए? Number 1, there is the requirement of ATP. Number 2, there is the requirement of enzyme. Number 3, there is the requirement of ribosomes. Number 4, there is the requirement of messenger RNA. There is the requirement of transfer RNA. ठीक है? और इसके साथ हमें और क्या चाहिए? We need magnesium ions. These are the tools or machinery. Next, then we will say we are proceeding towards the third thing, that is the process. Any process which is taking place, it has 3 steps. Number 1, initiation. Second step, the promotion, means continuation. And the third step is the termination. Right? So initiation. The protein synthesis initiates with the help of energizing the transfer RNA. Means ATP combines with transfer RNA, it forms amino acyl transfer RNA. Means that transfer RNA with AMP it is joining. Second thing, in the initiation step, it's a combining with amino acid, so the complex is formed AA, AMP, transfer RNA. Means उस transfer RNA के साथ amino acid भी जुड़ गया और वो energized भी हो गया. And all this process takes place in the presence of enzyme amino acyl transfer RNA synthetase. तो ये होगी हमारी initiation. Then we are moving how to promote this protein synthesis, means how elongation, promotion की बजाय मैं यहाँ पे word use करूँगी elongation. So इस elongation process में हम क्या देंगे कि ribosomes subunits, they are attached during the protein synthesis. The bigger unit will be having association with that energized transfer RNA amino acid complex. It will attach with the bigger unit. And in the smaller unit, messenger RNA polymerase will bring the messenger RNA. It will read the first anticodon present there so that it can bring that appropriate codon. And we know the first codon which is required as the initiator codon, that is AUG. तो AUG को लाने का जो function होगा वो किसका होगा? That will be of messenger RNA. AUG will, when it will be present, it is the duty of transfer RNA to see which anticodon is required so that there is the base pairing between codon. अगर AUG आ रहा है तो इसका मतलब AUG combine करेगा with UAP. उसके साथ ही ये combine करना शुरू करेगा. एक मिनट बेटे मैं कॉल का जवाब दे लूं. एक मिनट होल्ड करो. हेलो मैम, हाँ जी, तो ये कॉम्प्लेक्स बन जाएगा. सो जब एक कॉम्प्लेक्स, एक अमाइनो एसिड का कॉम्प्लेक्स दूसरे अमाइनो एसिड के कॉम्प्लेक्स के साथ ज्वाइन करेगा तो दोनों के ज्वाइन करने से क्या होगा? पेप्टाइड बॉन्ड फॉर्म होगा. फिर उनके साथ जुड़ेगा थर्ड वन, द सेकंड पेप्टाइड बॉन्ड विल फॉर्म. देन उनके साथ जुड़ेगा फोर्थ वन, then third peptide bond will form. When multiple peptide bonds are formed, the chain is termed as polypeptide chain, which is the another word for protein synthesis. Then I am coming to the third step, which is termed as termination. Means when this protein synthesis will stop. And this protein synthesis will stop when there are untranslated regions. In your NCERT, a word is given UTR. Untranslated regions are present on this messenger RNA, because whenever any of the one stop codon, UAA, UAG, UGA, any one of them, if it is carried by RNA polymerase on messenger RNA, transfer RNA बेचारे को पता ही नहीं चलता कि इसके साथ बॉन्ड किसका बनाऊं, वो बिल्कुल लॉस्ट हो जाता है. वो साहब हो जाता है जब बच्चा पेपर में पढ़ के नहीं आता ना, तो उसको पता ही नहीं होता मैंने लिखना क्या है. ट्रांसफर आरएनए का वो साहब है, उसकी बनती नहीं है बिल्कुल यूटीआर के साथ. So then there will be no bond and it will be the end of protein synthesis. So in this way, the protein chain will be formed in this process. It will be released into the cytoplasm, and all the necessary functions which are performed by these protein chain, it will start working. So this is all about protein synthesis. So this question may come in 5 marks. Then the diagrams, definition, machinery, process in proper steps with headings, it is mandatory to explain. Or it can come in chunks. इसके छोटे छोटे से पार्ट्स भी आ सकते हैं. राइट, क्लियर होगी ये बात? फाइन. ये बच्चे थोड़ा सा प्रोटीन सिंथेसिस से घबरा जाते हैं, पर घबराने की आपको कोई जरूरत नहीं है. when you will study it, definitely आप अच्छा करोगे इसमें. इसके नोट्स जरूर बना लेना. इसी तरह से डेफिनेशन, मशीनरी, प्रोसेस, उसके स्टेप्स, देन हेडिंग्स और डायग्राम साथ में. ये आपके पूरे जो है नोट्स बन जाएंगे. नहीं तो आई थिंक जो 24 अप्रैल से वी हैव स्टार्टेड दिस मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस, और इसके 12-13 लेक्चर्स थे. तो टिल आई थिंक 6th और 7th मई के वीडियोस जो मेरे हैं, they are very much based upon this protein synthesis step-by-step process, and so many good diagrams are there, because in NCERT the complex one is there. So those videos I have taken some of the content from Khan Academy also. So very good material and definitions are I have mentioned there, so you can follow that one. Or if you are having any reference book, जैसे वो हो गया आपके प्रदीप पब्लिशर्स वगैरह की कोई बुक हो, या ट्रू मैन पब्लिशर्स की हो, तो you can go through those books also. In fact, MTG, if you are having some of the notes which are given, they can be a ready reference for that. So you can get idea from there, right? और कोई हो आपकी प्रॉब्लम? अपना देख, अगर आपको लगता है कि इसको डाइजेस्ट करने में अभी टाइम लगना है, आई कैन स्टॉप क्लास हियर आल्सो एंड आई कैन टेक डाउट्स नेक्स्ट ऑन मंडे. बट इफ यू वांट मी टू डू एनी अदर, मोस्ट वेलकम, इट्स योर चॉइस.
मैम जीन टैक्सी क्या होती है?
क्या होती है बेटे?
जीन टैक्सी, वेक्टर टैक्सी.
स्पेलिंग बोलोगे?
जीन टैक्सी.
GENE, जीन, और
टैक्सी.
स्पेलिंग
TAXI.
TAXI.
XI.
टैक्सी मीन्स करियर, जो किसी के एक को दूसरी जगह से लेके जाता है. और किसको लेके जा रहा है? जीन को. हु आर द करियर्स ऑफ जीन? जीन किसके ऊपर प्रेजेंट होते हैं? व्हीकल्स कौन है जीन्स के?
BAC and YAC.
हाँ जी,
एक BAC और YAC.
बैक्टीरियल आर्टिफिशियल क्रोमोसोम्स एंड यीस्ट आर्टिफिशियल क्रोमोसोम्स, वेक्टर्स उसको कैरी कर लेंगे. इट इज गिवन इन योर DNA सीक्वेंसिंग ह्यूमन जीनोम प्रोजेक्ट, जिसमें आप वो पढ़ते नहीं हो सीक्वेंस एनोटेशन वाला, and expressed sequence tags, EST. So the two vectors are being used. So chromosomes, actually they are the vehicles of these genes, and the vectors who are carrying those genes, they are termed as the gene taxon. Anji, anything related to translation? Any questions? Shobhit, any question?
मैम लैक ऑपरेंट के बारे में बताना.
लैक ऑपरेंट, चलिए जी. लैक ऑपरेंट प्लीज याद रखना, बच्चों को ये टॉपिक आता होता है, कहाँ से वो मार खाते हैं? They do not understand what question is asked. Lac operon, first of all, it's an operon. Operon means is a set of the genes. Jacob and Manet, the Russian scientists, they have prepared a model to show that how the genes are present in proteins and how they are involved in the transcription and translation process. operon is the set of the genes, and what genes are present here? There are present structural genes, there are present operator gene, there is a present promoter gene, there is present repressor gene, and as well as there is present inducer gene. Now, lac is the short form of lactose, because in prokaryotes, this carbohydrate is present, the lactose is present, which is transcribed with the help of the enzymes into the simple sugars, right? And the enzymes required there are galactosidase, permease, as well as trans-acetylase. So इस लैक ऑपिरोन की दो तरह का ऑपिरोन होता है, एक होता है स्विच ऑन एंड अनदर वन इज टर्म्ड एज स्विच ऑफ. So there are three structural genes. One is called Z, another one is called Y, and third one is called A. Z is transcribing, means Z से जो अगला प्रोटीन बनता है उसका हम नाम क्या होता है? बीटा गैलेक्टोसाइडेज. Y is responsible for the formation of trans-acetylase, and A is responsible for permease. And it has one operator gene, it has one promoter gene, and it has here repressor gene, or you can say inhibitor I gene. This repressor is responsible for the formation of messenger RNA repressor. And whenever this repressor is formed, repressor always suppress, वो किसी को काम नहीं करने देता. This repressor, it has the ability to combine itself with the operator. Whenever it is combining with the operator, the RNA polymerase, which is required in this transcription process, it cannot make a bond with promoter. And when it cannot make a bond with promoter, ultimately what happens? These three structural genes, they fail to transcribe their प्रोटीन्स. मतलब उनकी ट्रांसक्रिप्शन नहीं होगी तो उनसे ये प्रोटीन्स भी नहीं बनेंगे. अगर प्रोटीन्स नहीं बनेंगे तो इसका मतलब है बॉडी में कोई भी सिंपल शुगर नहीं है. अब वो सिंपल शुगर कब बनेगी? व्हेन देयर इज अ कॉम्प्लेक्स कार्बोहाइड्रेट, एंड दैट कार्बोहाइड्रेट इज लैक्टोज, व्हिच इज एक्टिंग एज इंड्यूसर. जब ये अपने आप में ये इसको ट्रांसक्राइब नहीं करता तो इस टाइम पे इस लैक ऑपिरोन को हम एक नाम दे देते हैं स्विच ऑफ. बंद हो गया, इसने अपना वर्क नहीं किया, whatever it was supposed to do. Now here is present inducer, which is termed as lactose. This inducer, it's very powerful. Whenever it is present, it first of all makes a bond with repressor, and when it forms a bond with a repressor, the bond between the repressor and operator always breaks because inducer is present. So when this bond breaks, operator is free, promoter is free to make a bond with RNA polymerase. And when polymerase is present, in the presence of polymerase, transcription process takes place. As a result, the translation process takes place. Z, Y, A are responsible for the formation of these three enzymes or three proteins. And in this case, we are calling it as switch. If the question is asked simply, what is lac operon? What are the types? Explain with the help of the diagram. But you are supposed to give the definition. You need to mention switch off as well as switch on. वहाँ आपको डायग्राम भी दोनों की अलग अलग बनानी है, वहाँ आपको टेक्स्ट में भी लिखना है. It will be of 5 marks. But if the question is asked in a twisted manner, like Jacob and Maynard, they worked upon the lac operon. Lactose was used up by the prokaryotes, and after some time the system shut down. Explain or justify. अगर ऐसे करके इन्होंने लिखा है, it means they are asking you about switch off lac operon. Then in the text you can give about switch on कि inducer जो है वो repressor के साथ bond बना के operator के साथ इसका bond break कर देते हैं, and the process continue. When it is consumed totally, the lactose is not present in the nutrient medium, and as a result, messenger RNA is forming repressor, making a bond with operator, and afterwards the promoter cannot make a bond with the RNA polymerase. As a result, there is no transcription, there is no translation, and hence that operon is termed as switch off lac operon. So this is the reason when in the system lactose is consumed, the system shuts down. कई बार हम कहते हैं कि हमने तो सारा लिखा, हमें 5 में से 5 क्यों नहीं मिले, हमें 5 में से 4 क्यों मिले, हमें 3.5 क्यों मिले? Because examiner is looking forward if the twisted question is there, has the student understood? हम उस बच्चे को कहते हैं कि write down. अब whether तेरे को सिर्फ थोड़ा सा क्वेश्चन समझ आया, write down whatever is coming in your mind, where we feel the child has the capacity only to get the passing marks or just average marks. But if any student where we know the student has the stamina to score more than 95%, 90%, 100% marks, it has a stamina, 85% से ऊपर जाने वाला है. So those students, I always say, look towards the question, try to understand and frame your answer on the basis of the question. Got it? Shobit, is it clear, Vijay? So
Yes,
what
ma'am.
doubts you are asking, make sure at home, means you are at home. I mean, whenever you are free, you are opening your book. So please read those questions thoroughly, right? उनको अच्छे से करो. तो मुझे अगले डाउट्स भी फिर आप भेज देना. It's a 5:23 now, so I think I should leave you now. नहीं तो बहुत टाइम हो जाएगा, सेल्फ स्टडी का आपको टाइम नहीं लगेगा. So you can mention on the group जो भी आपने डाउट्स देने हैं. अगर ऑन द स्पॉट आपने मंडे पूछने, मोस्ट वेलकम. तो 11 बजे फिर हम मिलते हैं. मेक श्योर ऑन टाइम एंड एनकरेज योर क्लासमेट्स टू कम इन द क्लास एंड सो दैट ऑल द डाउट्स कैन बी क्लियर, राइट? और किसी का कोई अर्जेंट कोई ऐसा डाउट हो, यू कैन आस्क. नहीं तो मंडे आप मेरे को बता देना कैसे कैसे आपके क्वेश्चन है. ओके एवरीवन, फाइन.
Yes, ma'am.
Thank you very much, everyone. Have a wonderful day. So I am ending this meeting. Okay. Chaliye, enjoy your day. Have a nice weekend.